Have you ever wondered why some beetles glimmer like metal or why tropical fish shimmer with rainbow colors? These dazzling displays aren't created by pigments, but by light by itself. This phenomenon is called structural coloration, where microscopic structures bend and scatter light to produce iridescence. Look at this beetle shell. As we tilt it, the colors shift and shimmer. Now notice this tropical fish. Its scales seem to glow and change depending on the angle of light. What's happening here? Unlike pigments, which absorb certain wavelengths of light and reflect others, structural coloration works by manipulating light through microscopic layers. In beetles, thin layers of chitin in the exoskeleton act like stacks of tiny mirrors. In fish, layers of guanine crystals inside special cells called iridophores reflect and interfere with light. The result? Shimmering, angle-dependent colors that seem almost alive. But why evolve this? For some species, iridescence is camouflage, helping them blend into moving water or foliage. For others, it's a bold display for attracting mates or confusing predators. To understand how this works, we need to turn to physics, to one of the most famous experiments with light, the double-slit experiment. When laser passes through two slits, the light waves interfere with each other. Where waves overlap and sync, we see bright fringes, causing constructive interference. Where they cancel each other out, we see dark fringes, causing destructive interference. This interference pattern proves that light behaves like a wave. Now back to our beetles and fish. Their microscopic layers act like thousands of tiny slits or reflectors. When light hits these layers, it splits, bounces, and interferes, depending on the angle of light of how these layers are spaced. Some wavelengths reinforce, producing vivid colors. Others cancel, making those colors disappear. That's why a beetle's green can suddenly shift to gold, or a fish blue stripe can flash silver. Studies of beetles reveal multi-layered cuticles that act like photonic crystals, intricate designs sculpted by evolution to control light. Research on fish, like the neon tetra, shows how guanine platelets can even tilt like Venetian blinds, dynamically shifting color depending on the fish's needs. So next time you see a beetle gleam like metal, or a fish glow in shifting blues and greens, remember, what you're seeing is light itself, dancing through microscopic structures shaped by evolution into one of nature's most stunning tricks. Structural coloration is more than just beauty. It's physics, biology, and evolution woven together, a shimmering story of how life bends light to its advantage.